Myself, Dr. Mrs. Vijay Sethi. I have retired from the head of the department from the Division of Post Office Technology, IRI. Now, as you might be knowing that in continuation of the teleconferencing of, in continuation of the teleconferencing from the School of Agriculture from IGNOU, we are covering the course entitled Diploma in the Value-Added Products from Fruits and Vegetables, which deals with the course number 5, Food Microbiology. So food microbiology actually consists of 4 blocks, block 1, block 2, block 3, and total there are 13 units. Today we are covering 3 blocks. I am taking block 1, Introduction to Microorganisms, number 2, Controlling Microorganisms, Dr. Shruti Sethi will take, and number 3, Dr. Poonam Agarwal will be taking. Now I start with the Food Microbiology Journal. As you know that food microbiology deals with mainly the microorganisms, what are their main uses and their adverse effects. So I will take one by one. Next one please. I have to take myself. Now the contents deal with the definition of microorganisms, their classification, factors affecting microbial growth in foods, beneficial and detrimental effects, fermented foods and spoilage aspects. Now what are microorganisms? All of you know microorganisms are tiny organisms. They are very primitive, which cannot be seen with the naked eye. They are submicroscopic in nature, and they are our friends as well as foes. There are mainly 2 types of microorganisms, prokaryotics and eukaryotics. Prokaryotics are they are simple morphology and they don't contain membrane inclined with the nucleus, whereas for example bacteria, and others are eukaryotics. Morphologically they are complex, but they are with true membrane enclosed with the nucleus. Now they contain fungi mainly, molds and yeast. Now these bacteria, yeast and molds, you know that we're dealing one by one. Bacteria, they are the smallest one among these, smallest one among these, and they cannot be seen with the naked eye, I have told already. The shape of bacterium, they vary. They may be cocci, they may be rod shape, and they may be spiralum in nature. So many, as I have shown you in the figure, so many their shapes are there in there. And they may be motile or non-motile. Even their classification can be based on the motile, means they have motility, flagella is there, and non-motile, they lack flagella. Fungi, they deal with the yeast and molds. Yeast are bigger than bacteria. They are unicellular microorganisms like bacteria, and they are commonly egg-shaped, spherical, and some are of course cylindrical in shape. Yeast lack flagella, some are pigmented also, even bacteria also some are pigmented. On the other hand, molds are multicellular in nature and they are filamentous. They consist of hyphae, which are tubular-like structure, and this hyphae consist of mycelium, and they may also be colored and they may be produced by the fission. Bacteria may be produced by fission, where the yeast by budding and molds by spores. Now physiological classification of microorganism can be based on the temperature. On the basic temperature, we can classify them into 4 groups. Mesophils, minimum temperature 5 to 15 degrees centigrade, optimum 30 to 45, and maximum 35 to 37 degrees centigrade. Like we, for the human beings, like proper temperature, proper oxygen, proper nutrients are important for our growth and reproduction. Same way for the microorganisms also, they require particular optimum temperature for their growth and multiplication. The mesophils, they can throw on the ambient temperature, whereas thermophils, they are heat loving microorganisms. Minimum temperature is 40 to 45, optimum 55 to 75 degrees centigrade, and maximum 60 to 80. Can you put it in? Now psychrophils, they are you can say cold living microorganisms. Minimum is -5 to 5 degrees centigrade, optimum 12 to 15 degrees centigrade, and maximum 15 to 20 degrees centigrade. Whereas psychrotrophs, they can survive at low temperature. Their optimum temperature is more than the psychrophils. On the basis of oxygen also, we can classify these microorganisms into 4 main groups. Obligate aerobes, as the name suggests, they require free oxygen for their growth and multiplication. On the other hand, obligate anaerobes, they require very less oxygen or negligible oxygen for their growth and multiplication. Facultative anaerobes, they may grow in the presence and in the absence of the oxygen. Microaerophils, they can grow under low oxygen tension. In general, molds, they are aerobic. Yeast may be aerobic and anaerobic, that is facultative. And obligate anaerobes like Clostridium or Bacillus is aerobic. On the basis of requirement for water activity means the available moisture present for their growth and multiplication. Now, the bacteria, they require high moisture content because water activity 0.91, followed by yeast 0.88, then molds 0.80. Halophilic bacteria that can survive at high concentration of salt, and their water activity is 0.75. Xerophilic fungi, which contains low moisture content, and their water activity is 0.65. Osmophilic yeast is that which can stand a high osmotic pressure, whether it is developed by high sugar content or a high solid content or any solute, which cause the high osmotic pressure in the medium, so they can grow at that medium, and their water activity is 0.60. Minimal water activity means that this is the minimum, and above that they can go, but if you reduce them further, they cannot multiply. On the basis of nutrient degradation capacity, we can classify the microorganism proteolytic. Proteolytic means they degrade the protein. Lipolytic means hydrolysis of fats to fatty acids and glycerol. Saccharolytic, hydrolyze disaccharides, polysaccharides to simple sugars. Pectinolytic, they hydrolyze pectin. Even some of the bacteria, they can be classified on the basis of the staining. And like this I have told you, flagella, which are the organs of locomotion, even on that basis they can be classified, flagellate and non-flagellate. Flagellate means they have organ of locomotion, that is flagella is present, whether one flagella or both side flagella, that is different, 5 types are there. But on the basis of staining, there are 2 types, gram positive and gram negative. When you do with the basic dyes, gram staining dyes, these gram positive bacteria, those bacteria, they stain violet after gram staining. And if you counter stain them with the safranin, even then they will have that violet color. They will not pick up the red color in these cell walls, because these cell walls are mostly comprised of peptidoglycan layer, that is example Lactobacillus and Staphylococcus species. On the other hand, gram negative, they will not pick up the violet stain, and they will pick up the counter stain of safranin, that is they are red in color. The cell wall mainly comprised of lipopolysaccharides, example Pseudomonas species. Some bacteria, they have special features like capsule. When you see the cell, the layer of mucilaginous capsule is there, and the presence of capsules or slimy layer may account for the sliminess or ropiness of the food. Most capsules, they are polysaccharides of the dextran, lemon, and they serve as a source of the reserve material for nutrients and increase the resistance of bacteria under adverse conditions. On the other hand, the endospores, you know that vegetative cells are heat labile, where the endospores, they are heat stable, and our whole canning industry is based on the whether we want to destroy the bacterial spores or not, because vegetative cells are destroyed at 100 degrees centigrade. Bacteria of genera Bacillus, Clostridium, Sporicina, etc. have the ability to form endospores. And now endospores formation, they may be terminal, subterminal, and endospores are formed at an intercellular site and are resistant to heat, ultraviolet light, and desiccation. Lysis of the vegetative cell release the free endospore, which may remain dormant because they can remain under adverse conditions, they can stand that. And when the favorable conditions are there, they may germinate. Formation of cell aggregates, few of the bacteria, they clump together, or sometimes they are in chain, so it is difficult to kill them, and as compared to the vegetative cells, they will take more heat to destroy themselves. Now, classification of bacteria based on the nutrient requirement. This is very important aspect in the food microbiology. There are 2 types, autotroph and heterotroph. As the name suggests, autotrophs, they can synthesize their own food. Simple inorganic compounds like carbon dioxide, nitrogen from nitrites, nitrates, they grow on very simple medium. And further, they have been classified into two main groups, chemosynthetic and photosynthetic. Chemosynthetic means they can get energy from the oxidation reductions, and like carbon from carbon dioxide, nitrogen from nitrate, and examples are nitrobacter, others are nitrosomonas and thiobacillus, which this thiobacillus sulfur releasing the bacteria. Photosynthetic, they use the sun energy, sun rays, for their growth and multiplication as energy source, and examples are chlorobium and chromatium. On the other hand, heterotrophic organisms, this bacteria, they show considerable variations. For their requirement, nutrient requirement, they need organic materials for their growth. Again, there are two types, parasitic and saprophytic. Parasitic, you know that they are pathogenic in nature, which cause the diseases in human beings, like your E. coli, Salmonella, Clostridium, which are the main ones. On the other hand, saprophytic, they cause spoilage in the food, and they need minerals like sodium, potassium, magnesium for their medium for growth and multiplication. Examples are lactobacilli and protease, etc. Now coming to the yeast, I told you that yeast, they are generally asexual reproduction is by budding, and again main classification of the yeast is on the basis of spores, whether they are spore formers or non-spore formers. The yeast which produce spores, they are known as true yeast, and which do not produce, they are known as the false yeast. Now their names have been given to two yeast, like perfect yeast, sexual yeast, and other is ascosporogenous yeast or ascosporogenous yeast. The false yeast, on the other hand, they are imperfect yeast. They are also known as asexual yeast. They are called ascosporogenous yeast. Some of the examples for the true yeast are Saccharomyces cerevisiae, Ruxi, Pichia, and Sinula, and there are others also example. I have given here few ones, and then for false yeast, Rhodotorula, Candida, Tarulopse, Primatomices, etc. Now coming to the second unit, factors affecting microbial growth. Now there are main four factors. Composition, it comes under intrinsic factors, then microbial interaction, biotic factors, processing, disturbances, heat treatment, cleaning, sanitation, etc. Now the first three ones, they with the product quality, intrinsic factors, water relations, salt, sugars, pH, preservative, traditional natural like spices, herbs, fats, nutrients, etc. Coming to the second biotic factors, microbiota, then positive and negative interaction in food system. Processing disturbance like heat treatment, cleaning, sanitation, freezing, etc., or evaporation, high pressure, non-thermal processing, fumigation, and smoke. On the other hand, regarding the packaging and storage conditions, they come under the extrinsic factors, temperature, humidity, and then gas composition, like modified atmosphere storage, then controlled atmosphere storage, other gases, carbon monoxide, methylene, then active packaging, a packaging material, their permeability, and bio-based, and time of storage. Now we'll deal one by one. Coming to the intrinsic, which are food related parameters, pH, moisture content, or in other words the water activity. I have told you already that water activity deals with the available moisture present in a food material, which will be responsible for the microbial activity and physiological rate, etc. It is the bound water which is bound with the proteins, fats, other carbohydrates. It does not take part in the microbial activity. Oxidation reduction potential, then nutrient content, antimicrobial constituents, and biological structures. Now effect of pH. Most of the microflora, you know that this table you are seeing that most natural habitats deals with the pH 5 to pH 7, and if you go above 5, this causes the alkalinity. If below that, then it is acidity, towards acidic side. Then pH range of some common food items and pH range of some common food microflora. There are so many groups we can divide the food items depending upon the pH, but here I have give you few examples. Like citrus fruits, you know that they are acidic in nature, their pH from 2.5, soft drinks 2.5 to 4, fermented like beers they are 3.5 to 4.5, and meat, fish, etc. they are still higher, eggs, milk also come to the towards neutrality, and flours also above 6.2 to 7.2. Vegetables, it's a wide range 4.8 to 7. Fermented shock, this of course 10 to 12, this is still higher. Now coming to the microflora, this is I have given few examples, not the bacillus species. Even different molds, they will have the different pH range. So I have given the range, general range. For molds 0 to 11, yeast 1.5 to 8.5. You see molds have a wide range. Lactic acid bacteria 3.2 to 10.5, and these are the pathogenic, some of the pathogenic, they still have 3.2 to about 9, and bacillus cereus 4.7 to 9.3. So you can see that a wide range on pH they can grow. Now I was talking about the water activity. Now you may ask me, what is water activity? Water activity is the ratio of the water vapor pressure of the water in the food material itself by to that pure water at the same temperature, P upon P0. Now it is the amount of, I already discussed, amount of free water present in any food which is responsible for the microbial activity. In general, if you lower the water activity, I have told you that the activity will be suppressed to some extent. Water activity you can lower down by various means like drying, addition of salt or sugar, by freezing, because if ice crystal form there will be concentration of the solutes, so you can check them, by evaporation, then by hydraulic, some of the hydraulic gels also you can use to reduce the water, because actually the aim is to absorb the, bind the available moisture. Osmophilic microorganism, they can be called as a sugar tolerant microorganisms. They prefer high osmotic pressure which can be created by the high concentration of salt also, but generally the name for high concentration salt is given to the halophilic microorganism. Xerophilic microorganism which prefer low water activity, that is they require very less moisture content for their growth and multiplication. Now water activity of food materials and related microflora. Few examples have been given here. Pure water, this is one maximum water activity, or 100% you can say moisture content. Then you see here so many species have been mentioned. There are many more, but we have given few examples for that. Then human blood, lower than this 0.995, then seawater, then after that comes the bread, maple syrup and ham. All the fruits and vegetables generally come in this group also. Then salmoni, fruit cakes, jams, salted fish, cereal candy, dried foods. Because if your water content is more, then their perishability will be more. If water content is less, they will be stable. Shelf life will be more. Now, the interaction between pH and water activity controls microbial growth in food products. Now, you see here on the one hand, left hand side, we have given the water activity, and on the right hand side, there is the pH. You see, at high water activity and at neutral pH, high pH, most pathogens they can survive. Now, oxidation reduction potential, this is another important parameter for the growth and multiplication of microorganisms which cause spoilage. The oxidation reduction potential of a substrate may be defined generally as the case with which the substrate either it will lose or gain the electrons. When an electron or compound loses electron, then it gets oxidized. And while a substrate that gains electron, it becomes that it is reduced. Now, microbial growth in the food, when it is there, so the oxidation reduction potential also reduces. So this is sort of an indicator that how much microflora is there by finding out the oxidation reduction potential. The decrease in oxidation potential as a result of microbial activity is the basis of some long established rapid tests applied to foods, particularly in dairy products. Redox potential dyes are available, such as methylene blue and resazurian are sometimes used to indicate the changes in the oxidation potential, which indirectly correlate with the how much microbial load is present there. On the basis of redox potential, microorganisms have been classified as aerobic. You know, if aerobic means there the aerobic microflora will proliferate. I have told you they require particular oxygen. So if you reduce in the absence of anaerobic will thrive. And facultative microorganism, they may grow or they may not grow in the presence or absence of oxygen. Now modes, I have told you that they are aerobic. Most yeast grow best aerobically, but some are like wine yeast, they are anaerobic, and others also. Bacteria of different kinds, they may be aerobic, anaerobic and facultative also. Plant juices have an osmotic potential of plus 400 millivolt. It means that they are more of oxygen is there. So therefore aerobic bacteria and molds are the common cause spoilage of the products of this type. Now coming to the nutrient contents. Nutrient contents water, we have already discussed to some extent, source of energy, carbohydrates or other sources, source of nitrogen like proteins, then vitamins and minerals. They constitute the important composition of a food material that which type of microflora will thrive. Antimicrobial substances, some natural antimicrobial substances are present and which will arrest the growth of microorganisms. Coumarins, fruits and vegetables in some, lysozyme in cow's milk and eggs which will protect them. Then aldehyde and phenolic compounds, herbs and spices, like in mustard, allyl isothiocyanate has been reported, it has antimicrobial activity. Allicin, garlic, polyphenols, green and black teas, it has natural antimicrobial properties. Some biological structures, they are also responsible for the cause of spoilage or for their activity, metabolic activity. Now, you know that the fruits and vegetables, they are covered with the skin outside. So, naturally, I told you that microflora, that yeast, mold and bacteria, they occur everywhere in nature, whether it is soil, air, moisture, containers, fruits and vegetables. So, they are present. But the inner parts of the whole healthy tissue of the living, because it is enclosed, so here the microbial almost it is sterile and microbial content is less. On the other hand, grinding and mixing increases the surface area and they are more prone to spoilage as compared to their intact part. The shell on eggs, the skin on poultry, the shell on nuts and the rind on skin on the fruits and vegetables, they have surrounded the food, so they are less prone to the spoilage. Sometimes artificial coating is given, plastic coating or wax coating, like use shrimp packaging, where here the fruits and vegetables, they are wrapped with a polythene plastic films, so to check the respiration rates and microbial activity. Same way for waxing of fruits and vegetables, this film also protect from the microbial attack to increase the shelf life of the fresh fruits and vegetables. And sometimes natural wax is also present, like in some apples, some brinjals, so they protect them. Now coming to the extensive environmental parameters, temperature of storage, this is very, very important. See, if temperature is optimum for molds, yeast and bacteria, they are more prone to spoil. If you reduce the temperature like freezing, and then it will be slow. Relative humidity of environment, or we can say equilibrium relative humidity of atmosphere. Now this is also plays very important role. If humidity is high, condensation may take place and microbial attack will be more. On the other hand, if it is too less, moisture loss is there, microbial flora will be, inhibition will be there. Now presence and concentration of gases in the environment, oxygen or air. If oxygen is there, aerobic will proliferate. If less, then anaerobic. Carbon dioxide and ozone, you know, they have a inhibitory effect on the microorganism. They act as a preservative. Presence and activities of other organisms, lactic antagonism like bacteriocins which are naturally present, produced during lactic fermentation, then some of the acids like lactic acid and acetic acid vinegar and some of the aldehydes, they have antimicrobial properties. Now, I have told in the beginning that microorganisms are our friends and foes. Now they have three properties, the good, bad and the ugly. We'll cover one by one. The good means important of food production, provide better taste and texture. The bad means cause of foodborne illness, whether infection with the live organisms or intoxication with bacterial toxins. Ugly part is the food which microorganism which spoil the food with undesirable chemical changes, physiological changes. Industrially important microorganisms. Now, microorganisms in food and beverage production. They are used for the biomass production, yeast, microbial protein like single cell proteins, variety foods like fermented products which we will be dealing now, wine, beer, vinegar, sauerkraut, metabolite production, alcohol, vitamins and additives, organic acids, antibiotics. Then substrate conversion, steroids and wastewater treatment is also important aspect. Useful products from the agriculture materials. Now you see that where organic acids, several organic acids, then enzymes, polysaccharides, vitamins, amino acids, single cell protein and biological control agents, they have been some plastic, fibers, rubber, resin. Now this is diagram has been given that so many compounds can be useful products from agriculture material can be produced. Now chemical production through fermentation. Now through fermentation we can produce various vitamins as I have listed here, vitamin B1, B2, B6, B12, vitamin C, K3, biotin, folic acid, pantothenium, which are important our day-to-day life. Then amino acids, aspartic acid, crystalline cysteine, and many other which I have been listed here. Fermented foods, why fermentation is so important? Now, alteration of conditions for microbial fermentation such that pathogen and spoilage microorganisms are inhibited. This fermentation is brought about by bacteria, yeast and molds. Now, the generic process, now the upstream process is there, that media formulation is there, then inoculum development, then sterilization and inoculation. This is put in a fermenter, and after fermentation the downstream process is that product extraction is there, purification, then assay, waste treatment and byproduct production. So these are the few steps by which you can have this fermentation process. Types of fermentation process. There are three types of fermentation: solid state, extractive fermentation, submerged fermentation. In the solid state, as the name suggests, that free water is not available with the substrate. So fermentation is carried out as such. So this is known as solid state fermentation. On the other hand, extractive fermentation is carried out with the help of the enzymes. They are added, and here it is a very slow process. Of course, overall the rate of conversion of the substrate to the desired product is lowered, as against a submerged fermentation where free water is available and the fermentation is of course good quality, and such fermentation have been mostly to produce fermented food and beverage products. Now coming to the wine, all of you know, I think you might have tasted wine, beer, some of you, and mildly nothing. It is known as fermented grape juice. Now here the fermentation is carried out by wine yeast which is known as Saccharomyces cerevisiae. It is produced under anaerobic conditions, and the natural sugar present in the grapes, it is converted to the alcohol, ethyl alcohol, which is present in the wine mainly. In natural wines the alcohol content is 7 to 16%. Wines are produced, you can produce this alcoholic fermentation from any fruit juice, and that is, it will be named after that fruit. Suppose it is from berries, so berry wine. If it is from apple, it is cider, pineapple wine, like that. Now various types of wines are there. You might have heard few named: still wine, sparkling wines, dry wines, sweet wines, fortified wines, table wines, sherry, mead, vermouth, toddy. Because I will not go in detail one by one what are these types, I am just giving the few names which are very popular. Now this is a flow chart for the preparation of the white wines or the red wines. White wines mean they are produced from the natural grapes which are non-colored. On the other hand, red wines are produced from the red grapes. Now vinegar. What is a vinegar? Vinegar is a condiment which is produced by any fruit, any food material. First, there are two types of reactions. One is anaerobic fermentation, which is caused about by the yeast. I have told you alcohol. Then after that, this is oxidative fermentation. In the presence of oxygen, as alcohol is oxidized to acetic acid by Acetobacter acetal. By vinegar literally Translated as sour wine, it is one of the oldest method of fermentation used by man. Then it is the acetic acid produced by the aerobic fermentation, I have told you already, ethanol, and such gives the crustic flavor of aroma of the vinegar. The older the wine, better it is because it will have the good flavor. Now, it is mainly used, I have told you, but acetic acid as such is a very good preservative. Vinegar pickles you might have heard, so you can use 4% acetic acid. Sauerkraut. Sauerkraut in foreign countries, it is very popular. Now, this is prepared from the fermented cabbage, shredded cabbage, and here the fermentation is carried out by 2 to 3% dry salting. The natural microflora, it can be prepared either by the under controlled conditions by pure culture fermentation or by natural fermentation, and the end product is lactic acid, and this lactic acid is a very good preservative. Now, anaerobically carbohydrates undergo decomposition like alcoholic fermentation, lactic fermentation, coliform type fermentation, propionic acid fermentation, butyric acid. Now, here we have listed the fermented food products from fruits, vegetables, beans and related substrates. And the first we are given the which food, then the ingredients, then the microorganisms involved and which country they have been produced. Now, microbial utilization of food processing industry waste. We can prepare so many products by fermentation, microbial fermentation, ethanol, biogas, single cell protein, waste acids, baker's yeast and animal feed, etc. Probiotics, they are useful to organisms that colonize our body and they are good for prevention of the colon cancer and detachment of cholesterol plaque from arteries. Now, pathogens, I have told you already, they are toxin producing, they cause diseases. Then come the last part of spoilage of food. Now, here we have shown some spoilage and then fermentation that also cause. There are mainly two types of spoilage, abiotic spoilage and biotic spoilage. Abiotic means from chemical changes, cause different types of chemical changes in the product, hydrolysis, action of enzymes, oxidation of fats, putrefaction of proteins, browning reactions, etc. Physical changes also occurred here. Then temperature control is the major factor to provide longevity of the fruits and vegetables. Biotic means which have by microbial action. Then here we have shown the spoilage in the low and medium acid foods which have pH more than 5 and pH less than 4.5. You see here either they can be spoiled by thermophils or mesophils, again by aerobic or anaerobic. Same for mesophils. And coming to the spoilage in acid and high acid foods, bacteria, yeast and molds are involved, and mesophilic and thermophilic bacteria, they may be spore formers or non-spore formers. Then classification is based on perishability. How long we can store the fruits? Three classes are there: perishable, semi-perishable and non-perishables. This is the perishability of foods. We have shown here by diagram which are perishable and which are non-perishable, or you can say semi-perishable. Principles of food preservation is to check the spoilage. We can do by three ways: delay or prevention of microbial decomposition of food, delay or preventing of self-decomposition of food, and prevention of damage of the food caused by the insect, rodents, birds and mechanical cause. Factors governing spoilage. This is very important aspect. Number, kind and stage of growth of microorganisms. If higher, then more spoilage. If lower, then it will prevent the spoilage. Temperature, I have told you, optimum storage temperature also leads to the spoilage. Then suitability of the food material, by the nutritional quality of the, then physical state, whether you have heated it, dried it, frozen, the moisture content. Then coming to the pH of food, we have already discussed. Then foods having pH less than 5. This is the demarcation line for the pressure crystallization and atmospheric crystallization of the food material. Presence of air, aerobic and anaerobic microorganisms will grow depending upon the present in the package, how much oxidative changes will take place, and that may lead to also microbial flora. Changes associated with the food spoilage, there may be putrefaction, then nitrogenous compounds like amide, urea and others, they decompose ammonia, carbon dioxide and other products. Hydrolysis of the polysaccharides will lead to the monosaccharides and they ultimately will be, or glucose will be oxidized to carbon dioxide and water. Then ethyl alcohol is oxidized to acetic acid. We have talked about in acetic acid vinegar. In general, I can say physical and sensory qualities of the food also undergo changes, thus making the product unfit for human consumption. Spoilage of the canned foods. Now, there are mainly 4 types, but microbial spoilage is most important. There is a microbial, chemical, physical, and miscellaneous. In microbial, if you do under-processing, even then it is spoiled. Inadequate cooling, post-process spoilage, that is leaked through seams, and pre-process spoilage, that is autosterilization, or when you keep the, after cutting the material, when you keep it for longer time before canning, then there may be spoilage. Chemical, mainly hydrogen swell is caused, because if the acid foods are there and it will react with the tin plate, it may cause the spoilage. And various types of swollen cans are there. They may different stages, flipper, springer, swell. I will not take in detail. Physical means faulty retort operation, under exhausting. Exhausting is very, very one of the important parameter in canning industry. Overfilling and pendling. Miscellaneous, rust, damage or denting, etc. Now, controlling microbiological quality of food. We must focus on the food itself, the people involved in handling and the food, the facilities and equipment. Good hygiene and sanitation will prevent foodborne illness breakouts, maintain customer goodwill, and maintain the quality of standards. See, it is not only the nutritional quality which will say that is important to retain, but this is the microbial safety, food safety also, which is of course related to the microorganisms which spoil the food. Now, in the end, I'll thank all of you. Thank you. We can summarize that you might be knowing that microorganisms, how they are important our day-to-day life and how we can prevent them. I told you that on the perishability of the food material, you can classify into different groups. So how can you do? See, you can control them. There are various ways of controlling a microorganism. One is by heat, by thermal processing. Either you do pasteurization or sterilization in the canning, and second one you can do at low temperature by freezing, because what happens that during freezing ice crystal formulation is there, so available moisture is there and temperature is also low. So if temperature is low, the microbial activity will be arrested to some extent, their enzyme activity will be slow. And the other is that you can reduce them by increasing, like jams, jelly, you make various products. So here the principle is that high sugar content, they act as a preservative. Same way in the pickle, high salt content, it acts as a preservative. Now coming to the natural, like I told you, bacteriocins. Bacteriocins are the compounds, proteinaceous compounds which are formed during lactic fermentation. I told you various lactic acid bacteria are there naturally present, so they will bring the changes in the food material. Now in sauerkraut preparation, you are not changing the identity of the material. As such, kanji, you see, on the other hand, this is also lactic fermentation. Kanji is very popular in north, but you see their juice is fermented, and this especially in summer, this is a cooling effect. But in the sauerkraut, identity will remain same, and there is a lactic acid. Lactic acid is very good for our digestive system, microflora. Even you take curd, that also contains the lactic acid. So when you want to increase the shelf life of the product, not only you have to see the nutritional quality, but you have, on the other hand, you have to see the microbial safety also.